Good morning. Thank you for joining us for our latest Department of Defense Information Analysis Center webinar that will cover DARPA's Biostasis program. My name is Steve Rutherford, and I'm the director of the HDIAC. It is my privilege to host Dr. Tristan McClure-Begley for today's presentation. I want to start off by thanking all of you and saying that I'm grateful to everyone who's taking time out to join us in the middle of this holiday season. I know there are lots of demands on all of our time, so thank you for spending an hour with us today. I'm very appreciative of it. I'd also be remiss if I did not thank our sponsor, the Defense Technical Information Center. If you've not checked out DTIC's website, you can find it at www.discover.dtic.mil. And you should take a moment to review their expansive services. There's a lot of offerings there. And without DTIC's sponsorship, this webinar series just wouldn't be possible. So I want to kick it off with a few admin notes as we start out today. Note that all your lines have been muted, so if you have any questions during the webinar, you can submit them using the attendee chat box that will be on the left-hand side of the screen. We'll work to save the last 10 minutes or so of the presentation to go over those questions and discuss them and give Dr. McClure-Begley a chance to answer. Please also note that this webinar is being recorded, and a link for the recording as well as the slides will be available at the HDIAC website for later download for our members. With that, it's my pleasure to introduce you to today's presenter, Dr. Tristan McClure-Begley from the Defense Advanced Research Project Agency. Dr. McClure-Begley came to DARPA from the University of Colorado Boulder, where he was a research assistant professor in the Department of Molecular, Cellular, and Developmental Biology. His academic studies focused on molecular mechanisms of perturbation of complex biological systems, particularly drugs of abuse, toxins, and neurodevelopmental disorders. Prior to his faculty appointment at Colorado, Dr. McClure-Begley was a postdoctoral fellow in the Department of Psychiatry at the Yale University School of Medicine and is an alumnus of the Yale National Institute on Drug Abuse Neuroproteomics Center. He joined DARPA as a program manager in October of 2017, and his scientific pursuits at the agency involved novel chemical biology approaches to treating disease and injury and developing methods to accelerate and protect learning and executive functions. Dr. McClure-Begley earned his Doctor of Philosophy in Integrative Physiology and a graduate certificate in Behavioral Genetics from the University of Colorado Boulder. And with that, it's our distinct honor to host him today, and I will turn it over to you, Tristan.
All right, thank you, Steve, and thanks to everybody at the HDIAC for their assistance in scheduling and executing this particular seminar. I think I will just launch right into the topic of discussion with a brief background and some biography on the DARPA Biological Technologies Office. So DARPA was created originally in response to the launch of the Sputnik manned space venture. That term, the Sputnik moment, is directly relevant to the creation of DARPA, which as an agency, its core mission is to protect from and to generate technological surprise. And since its inception, the agency has been directly responsible for a vast array of highly disruptive and penetrant technological innovations, such as the internet, global positioning systems, unmanned vehicle navigation, stealth technology, microelectronics, and now most recently, the launch of the Biological Technologies Office in 2014. Now, the creation of BTO marks the first new technology office in DARPA in more than a decade, which highlights the importance of biological science discovery and the application of biology to national security with DARPA's investment in this field. The early BTO programs focused on three major areas: outpacing infectious disease, development of neurotechnology, and synthetic biology, with programs that made early investments in technology such as gene-encoded antibodies, brain-machine interface for advanced prosthetic control and haptic sensorization, and biomanufacturing. Now, subsequently, the office added areas of interest such as advanced diagnostic platforms for early and rapid detection, and an emphasis on warfighter readiness and recovery, which is right about where I came on board. And prior to being a DARPA program manager, I was a DARPA performer. So I came to the agency, as Steve mentioned, from the University of Colorado, and I was there involved with a DARPA program called Rapid Threat Assessment, which was all about a fast identification of molecular mechanism of action for unknown or novel chemical threats. It was a very large multidisciplinary effort involving quantitative mass spectrometry for proteomics and metabolomics, RNA-seq, multiplex cell activity monitoring, all in the pursuit of platform technology that could help shorten the gap between the discovery of a novel toxin or drug and our understanding of how it worked. Now, as that program was drawing to a close, I was recruited to DARPA to help craft and execute new research efforts in the space of novel therapeutics to address persistent health concerns in the active duty and veteran military population. So as Steve mentioned, my academic training is largely in physiology and pharmacology, so I gladly took on the challenge of finding new ways to intervene in situations where physical well-being is threatened. And chief among those concerns, and the topic of our discussion today, is going to be what to do about biological systems of collapse following injury or exposure to environmental stress. So the inception of the Biostasis program, is largely addressing a capability development effort. The Department of Defense has operations that are distributed around the world, and all of which are dependent on the health and the support of its service members. As such, there are circumstances and environments where health and logistics infrastructure is not sufficient to intervene during these dynamic and stressful conditions. A major limitation with all biological entities, whether we're talking about biomolecules like vaccines or antibody products or whole organisms, all the way up to human beings, is that there is a tendency for them to lose functional capacity over time. And when challenged with inhospitable environments like extreme temperatures or reduced hydration, following acute damage like traumatic injury, the duration of functional capacity can collapse very, very quickly. And once that biological system's function has been perturbed with significant strength, there's a race now to intervene before progressive, irreparable damage of the system takes place. And with this challenge in mind, we created the Biostasis program to explore how some fundamental biochemical interactions can be used to inform an engineered applied solution to this problem of biological systems and products losing functional capacity over time and under stress. This program is first and foremost a fundamental research effort, and I should stress that up front. While we are certainly motivated by the need to design and demonstrate therapeutics, Biostasis is not itself an advanced development effort that is working expressly to bring products to market in the near term. Rather, we are testing fundamental hypotheses about what is possible in this space according to experimental evidence for what we hope will eventually become an applied technology with real-world value. And with that in mind, there is a pressing need to address these issues. With the changing nature of armed conflict around the world, the U.S. military may well find itself in scenarios where prolonged field care of injured soldiers and civilians becomes a much more common circumstance and paramount concern, and where logistical burden to the fielding of cutting-edge therapeutics needs to be circumvented in order for us to reap the maximum therapeutic benefit. And part of what we hope to position Biostasis technologies to address are, practical solutions to the kind of field-forward emergency medical care that could become likely when encountering circumstances like near-peer adversaries and the type of casualties that could potentially result from these conflicts. So why is it that we, as the Department of Defense, care? Here are some examples of how time represents a contributing factor in military medicine and support. In the panel directly to the left, you'll see that there is this pattern, what is often referred to, that defines the relationship between elapsed time since critical injury and the risk for incidents of death. And it's called the trimodal distribution of death following trauma. And it collects the majority of terminal cases under 3 peaks. The first is represented by deaths occurring within the first seconds to minutes, which are usually attributed to penetrating trauma and massive hemorrhagic shock. The second peak occurs in the range of minutes to hours and is caused generally by persistent airway obstruction or uncorrected blood loss. And then the final peak occurs in the days to weeks range and is usually attributed to infection or cumulative irreparable damage and organ system failure. Now, this distribution is particularly striking in the context of a field-forward battlefield scenario where physical distance with respect to adequate medical infrastructure is a major confound. It has been estimated that up to a quarter of all combat-related deaths in Iraq and Afghanistan were, quote, potentially survivable had they occurred in a region more similar to, say, a stateside urban environment where most injuries occur within a reasonable radius to a hospital. And as such, the adage, delays result in death, is highly appropriate here. Traditionally, our interventions on the battlefield to deal with these scenarios have been to, first of all, accelerate medical transport. How do we close the gap in terms of time by accelerating our ability to transit distance? And to assemble agile medical treatment units, essentially closing distance, speeding up transit to try and better enable what is often referred to as damage control resuscitation. And generally, this process seeks to stop bleeding, replace lost blood, and then prevent the triad of death, which consists of coagulopathy, acidosis, and hypothermia. There have been some investments in tissue replacements like blood storage systems and blood products that can be administered to provide some degree of physiological support. But clearly, there are innovations that can be brought to bear in these circumstances. Now, speaking of blood and blood products, biological therapeutics and reagents, exemplified in the middle panel here, are particularly prone to loss of function over time and under stress. Whole human blood, which is, truly can be a life-saving intervention under these field-forward circumstances, it has a limited lifespan. And even when stored under controlled conditions, is only good for about 35 days. And I think many critical care practitioners would probably generally prefer to stay away from blood that is toward the end of that range. Now, in the military health system, particularly for field care, blood availability is challenging given the time frame that is already devoted to collection, processing, and transport, meaning that for many operations, as much as 2 weeks have already passed by the time the blood is actually on hand in theater and can be used. So the actual field lifespan of that particular product is substantially less than its entire duration. And much like whole blood, many highly valuable treatments such as vaccines and therapeutic antibodies require careful temperature and humidity control. I mean, look at the scenario we're in right now with we have traditionally relied on expensive and cumbersome cold chains to protect the functional capacity of these products. But again, refrigeration and climate control is a power-hungry process. And such power does not exist everywhere that we need these things to be. And then lastly, in the panel on the far right, looking at the rapid onset of infection and illness that often accompanies serious injury, the onset of sepsis can often outpace the therapeutic action of an antibiotic administered, up front, even with a loading dose. Careful patient monitoring is useful to track mounting concerns, but again, sometimes the cascade from subclinical presentation to life-threatening illness can occur very quickly and leave clinicians with little resource. So, to that end, in the Biostasis program, we are tasked with developing novel chemical interventions that can reversibly slow or pause biological processes and systems while preserving functional capacity. Taken together, we are seeking to develop new classes of therapeutics operating according to a novel therapeutic mechanism in order to lengthen the duration following damage or stress where recovery after intervention is possible, literally trying to slow life in order to save life. Now, in order to address these very pressing acute care and logistical issues, we asked a very fundamental question, which is how can we get a complex biological system to slow down in such a manner that it prevents an accelerating functional collapse? Coupled to that notional question is the more practical one of, will such an intervention actually be therapeutically useful? Now, in order to answer these applied questions, there are some guiding principles and additional fundamental questions to ask. So the concept of biostasis has its origins in a very basic question. and that is, what is the relationship between structure and function that enables life to persist? This is almost an echoing of the Bauhaus art movement principle of form follows function. We think of living things as always in motion or active to some degree. Neuroscience, for example, many concepts are explicitly understood in terms of patterns of activity, such as synaptic plasticity and the formation of memory. But some of the information stored in living things is structural, such as in genes, where explicit sequences of DNA base pairs are organized in a two-dimensional series, as well as in three-dimensional conformations. And the activity of cellular components is highly dependent on structure. Protein conformation arises from contributions of amino acid sequence, post-translational modification, folding, etc. One of the questions I repeatedly pondered is, what is the nature of the relationship between continuous process and structural element that promotes the adaptive and resilient nature of living things? In other words, since organisms are non-equilibrium systems, it's curious to interrogate what are the relative contributions of continuous flux and activity and three-dimensional structure to that living thing. It's a question that almost touches the edge of a philosophical notion, because life is a set of processes acted out with and within physical objects. So what are the relative contributions of form and function to phenotype? And one interesting way to contextualize this notion, and one that ultimately gave rise to the core hypotheses being evaluated in biostasis, comes from the consideration of what it means in the natural world to pause or slow the execution of life activities. There are fantastic creatures distributed across taxa that are capable of reducing their existential functions to either minute levels of activity, such as in hibernation or torpor, or even to states where almost all outward signs of activity are absent, which is collectively referred to as cryptobiosis. And these organisms are not super restricted to a single phylum either. Cryptobiosis and biological slowing writ large can be observed as complex traits arising independently multiple times. And with that in mind, it's important to consider what are the fundamental biochemical attributes that these taxonomically diverse yet phenotypically similar traits have in common. The discrete molecular effectors of a cryptobiotic process and the mechanisms through which they act represent a significant focus of the work being pursued in biostasis. Some effectors may be homologous proteins arising from an evolutionarily common ancestor, while others may be homoplasies arising independently but with similar functions. And once we distill what is understood mechanistically from this panoply of examples, what can we actually do with it? So to that end, one of the first things we started with during the inception phase of the program is to identify common mechanistic components underlying the phenomenon of cryptobiosis. And what we came up with is essentially the control of conformational flexibility and hydration of proteins and macromolecular machinery inside cells. So applied to the known or inferred mechanisms of cryptobiosis, it seems to fit reasonably well. The North American wood frog, for example, Lithobates sylvaticus, is a fascinating amphibian which can survive winter without hibernating by literally freezing. There is ice inside of that animal's body cavity. And wood frogs have a complex set of physiological responses to plummeting temperatures. And a key aspect is that they actually have mechanisms where they pack their cells with the disaccharide trehalose, which helps crowd the interior, displace water from the surface of proteins, which increases the local viscosity, which in turn slows protein conformational flexibility and thus catalytic activity as water is cooling. This results in a state of freezing that is actually tolerated by the cellular machinery and can resume function once the water returns to a more life-conducive temperature, if you will. Now, other cryptobiotic organisms, like tardigrades, through various mechanisms, including a number of combinations of upregulated metabolites and upregulated disordered proteins, they act as both molecular crowders that can facilitate phase transition to a gel-like state, as well as chaperone proteins to stabilize components and machinery. Again, the critical factor to keep in mind here is that these organisms undergo all of these relatively large-scale changes in the composition of their cellular contents. And the cold tolerant systems benefit from the change in enthalpy introduced by the low temperature. But it's really the loss or the phase change of water and the resulting decrease in cellular energetics, including enzyme catalytic activity, mechanotransduction, et cetera, that is consistent between these different models in the induction of the stasis state. At least that's what we've observed up to now. And there is a bit of chicken or egg when it comes to the relationships between protein chaperoning and cryptobiosis. For example, in freeze-tolerant organisms, the cells are protected by these various molecular effectors that allow them to resist damage due to low temperatures and the phase changes of water, like the formation of ice crystals. One can argue that in those cases, it's the cold that is really the source of stasis, since temperature is a factor in chemical reaction rates. And since, radically simplified, life is a set of chemical reactions, slowing them down is what is slowing down the biology. But there are other types of cryptobiosis, such as the organisms that can survive extreme desiccation. And in those cases, we refer to that process as anhydrobiosis, or life without water. And here, the metabolic process normally associated with living things is also paused, but the temperature dependence is much different. Now, interestingly, some of the same molecular effectors that are known to be good cryoprotectants for biomolecules like trehalose and other disaccharides and the novel proteins discovered in tardigrades are good protectants for anhydrobiotic states. A line that one can draw through these processes is essentially that the notion of controlling the conformational flexibility and catalytic activity of the proteins by manipulating their interactions with water. So part of the fundamental question we're asking in biostasis is how much biological stability can be reached by virtue of crowding and chaperoning alone? And tethered to that fundamental question is the more applied notion of, well, can a quasi-static state, reversible according to the laws of mass action, be leveraged for therapeutic purposes, whereas an unchecked system is careening towards collapse? Now, these questions, in the context of the fundamental hypothesis at their core, addresses a major challenge within the biostasis program, and that is the issue of scale. If our core hypothesis is valid, and chaperoning writ large is sufficient to stabilize biomolecular constituents of complex functional processes, will the actual intervention scale with both the physical size and the complexity of the system? And something else I wanted to address programmatically in biostasis is to avoid what I refer to as the crypto-fab sine wave, where a molecular fundamental aspect of a cryptobiotic process is discovered, it gets reduced to an essential single element and then applied to a different system with the expectation of identical results. And then interest falls off when those results aren't as stunning as hoped or promised until the next iteration comes along with a similar reductive logic and a similar lack of translatability. This has happened a few times, hence my analogy of sine wave. And it's important to appreciate that wood frogs don't just survive freezing as they overwinter because they can make trehalose. They need to import it into cells in order to achieve a high intracellular concentration. They need to induce alterations in complex physiological processes like heart rate, vascular tone, et cetera, such that as the temperature drops, things come to an orchestrated, reduced steady state or outwardly observable pause for maximum durable protection. And that process didn't involve the evolution and modification of just one novel protein or a sequence of enzymatic activities to produce a single novel metabolite. It required subtle changes distributed across that organism's genome and by consequence involving many coupled processes. It's a big ask to get an organism that didn't evolve a certain characteristic or function to suddenly accept that stimulus and assume that state, which brings up the next major challenge, which is translatability. How do we confer a functional state, or really a reduction in a complex set of functional states, to an organism or tissue of an organism that never evolved those traits in the first place? And this means the real trick the biostasis program has to pull off is figuring out how to deliver these capabilities in as few steps as possible to a system or a product that is not operating with the benefit of millions of years of evolutionary guidance without re-engineering that system. So we are specifically pursuing approaches that can act on the system without needing to re-engineer it at the genetic level. I wanted to avoid introducing distributed chimeras throughout the system in order to facilitate the impact of treatment. Complex systems respond very differently to perturbations when the source is genomic versus chemical. Think of differences in response to RNA interference knocking down protein expression of an enzyme versus adding an antagonist of that enzyme. One perturbation to the system is hard enough to study with respect to establishing causality and elements to be optimized for function. Introducing such things into a background resulting from genomic modification would make deconvolution of the effects almost impossible. So to that end, and in keeping with the theme of having some degree of target product profile available from the start to guide the experiments and their proposed interventions, we want the biostasis therapeutics to be passively delivered to the recipient, not requiring ectopic expression or the hijacking of cellular translation machinery in order to produce the actual active components. So with that knowledge in mind, one, the fundamental mechanism we're exploring is if reversible molecular chaperoning is sufficient to induce and maintain a viable static state and is scalable across systems of escalating complexity. And two, any biostasis interventions must be administered passively to the system. Genetic manipulation is off limits, and such products must be made in a fashion that their stasis-inducing effects can be readily reversed, and we can transition to discuss the approaches being pursued. So, coming up with unifying theory was a big part of program inception and how it is being managed and kept focused for its duration. Another big part was identifying technological advances that enable the realization of this desired capability. So based on recent advances reported in the scientific literature and our own technical expertise, we identified three major areas of innovation that we felt could be applied to this problem. First is pharmacological chaperones. Next is intracellular polymers. And last are engineered disordered proteins. So first, we describe the use of chemical chaperones to stabilize protein conformational flexibility. So vastly oversimplified, most biochemical reactions that enable life rely on proteins for catalysis in some fashion. Most chemical reactions occurring in biology are thermodynamically disfavorable. They won't occur on their own. So you need catalysts to help chemical reactions happen that otherwise wouldn't occur spontaneously. Proteins catalyze reactions by virtue of their ability to adopt different shapes and move in 3 dimensions. There are also forms of cellular stress caused by energetic demands in assembling proteins and protein complexes and ensuring that they achieve and maintain the proper folding patterns for stability and activity. So there are examples of drugs and other small molecules that exert therapeutic effects due to their ability to bind and help fold proteins that are improperly folded, often due to mutations in the primary amino acid sequence, or to help relieve cellular stress due to improper protein folding at the level of the endoplasmic reticulum and its export machinery. These drugs are called pharmacological chaperones and have been implicated in some interesting biology for a while now. So for example, one of the most effective therapeutics for cystic fibrosis is actually a chemical chaperone that rescues the misfolded CFTR protein. It's been hypothesized that binding and stabilizing intracellular targets of psychiatric drugs is also a contributing mechanism to their therapeutic activity by stabilizing receptors or transporters against degradation and reducing the energetic cost while bound. There's a number of pretty decent reviews written by a colleague of mine at Caltech, Henry Lester, on this topic. The problem with a lot of pharmacological chaperones is that they have only a few targets that they interact with because they bind rather specifically and selectively. So what we want to find as part of the biostasis program are kind of the opposite extreme, right? Rather than a client-restricted chaperone, we want to identify promiscuous pharmacological chaperones, small molecules that can interact with and stabilize lots of different proteins with at least approximately equal potency and efficacy. A key here is that it has to be reversible. There are plenty of molecules out there that can bind every protein and lock it into a restrained conformation, because that's how formaldehyde works, and that's not what we're after. What we really want are classes of molecules that can induce a stabilized conformational state in multiple structurally and functionally diverse client proteins, and then test those compounds to inform structure-function relationships that guide elaboration and eventual evaluation as adjunct therapeutics. Another approach is extending that notion of intracellular crowding, typified by the mechanisms in the wood frog. Getting the interior packed with some form of solute to constrain functional flexibility and macromolecule diffusion, preventing activity, while also providing an environment that helps prevent critical nucleation events. That's a challenge. I mean, we're, usually use the analogy of packing peanuts in a shipping container or trying to get from one end of a crowded train car to the other. There have been outstanding advances in biocompatible polymers lately, including some that can be assembled inside cells and have been shown to act as a sort of molecular sieve that can restrict mobility of large molecules like proteins while permitting diffusion of smaller metabolites. And I think it will be fascinating to see if we can actually engineer material that can be taken up in monomer form, much like the wood frog cells import trehalose, and then be induced to form a uniform intracellular gel and lock constituents in place, effectively arresting much of the cellular machinery, but then be reversed without harming anything. And then our third innovation is in designer peptides. The discovery that tardigrades actually induce the upregulation of some proteins with very interesting characteristics was a big inspiration for this area of investigation development. The tardigrade experiments detailed in a publication by Thomas Boothby et al. In Molecular Cell in 2017 describe the ability of tardigrades to survive extreme changes in environmental conditions, provided the transition didn't happen really fast. So tardigrades that were slowly frozen or slowly desiccated would assume a particular state called the ton state that was accompanied by the upregulated abundance of a number of interesting proteins. The tardigrade proteins have traits and amino acid sequences associated with a high degree of disorder. That is, they don't readily or neatly fold into predictable 3D shapes. Instead, they're kind of floppy. And when the tardigrade is stressed, it makes a lot of these disordered floppy proteins, which it turns out probably serve a number of functions in the cell. For one, they're made at great quantity. So you could imagine that they're helping to, first of all, crowd the interior of the cell. But they also bind to other proteins and act as chaperones, further aiding the protection and facilitating decreased catalytic activity. Key to our inspiration for pursuing this class of interventions was the observation in the Boothby et al. Publication that even individual tardigrade disordered proteins, when transferred into an unrelated organism, like a yeast, improved yeast desiccation tolerance when ectopically expressed, and could also protect enzymes from denaturation by heat and dehydration. So a lot of progress is apparent in the literature on protein engineering using completely physics-based approaches that model protein structure according to the physicochemical properties of amino acid side changes and charges, and can simulate their 3D shapes and possible functionalities accordingly, as well as evolutionary approaches that relate shape and function based on similarity in sequence and anticipated shape due to the distances and connections between residues in the chain. So given the distribution of stasis-enabling proteins scattered throughout the extant taxonomy, such as proteins produced by early embryonic states of organisms that enable stress tolerance, proteins that protect seeds from desiccation, or brine shrimp from hypersalinity, et cetera, there is certainly room for applying those methods to this problem. Can we derive enough structure-function relationship knowledge from the existing structural information about the organisms that utilize these resistance-enabling proteins to help find design principles and then apply them to target molecules and biological systems of interest for durable stabilization? So this is ultimately how we structured the program. If the ability to just push pause on a complex biological system is the main course, this program is really more like setting the table. We are building the tools and seeing how close we can get to that type of intervention in progressively more complex biological model systems. There is certainly the appreciation that much fundamental biochemistry, structural biology, synthetic organic chemistry, and cell biology all need to be brought to bear on this problem. And to do so, we broke the program into two major technical tasks. Task one is the front-end design process, where exemplar compounds are designed and optimized for activity according to the guiding hypotheses and the core technology of the approach, right? Whether it's small molecule, biocompatible polymer, or engineered protein. Task two is all about showing activity in the relevant biological model systems. So as I mentioned previously, with the major conceptual challenges that need to be addressed in the program, we're not ignorant to the fact that scale and translation are critical. To that end, we designed the program to roll out with intermediate goals and applications along the way. So early on, we're exploring the ability to safely constrain and stabilize simple biomolecules like enzymes. And one of the obvious applications for biostasis products is the ability to store and transport valuable biomolecules like therapeutic enzymes, vaccines, and antibodies without a cold chain. You know, keeping things cold is really expensive and often really difficult to do in environments when you might really need those things. So being able to formulate such products for improved shelf stability or even just tolerance to temperature fluctuation can thus be a major improvement in logistical capability. In the subsequent phase, we ramp up to stabilizing cells because, again, obviously, being able to carry blood or blood products for ready use without needing to keep it cold or from a fresh donor is a very attractive capability for both military and civilian critical healthcare practitioners. And in the end phases of the program, we are looking for stasis effects on complex biological systems, complex in terms of both cellular heterogeneity as well as structural heterogeneity. Organisms that represent multiple levels of physiological complexity that can be reversibly slowed and show therapeutic activity from such an intervention. Now, right now, the program is just about midway through phase 2. So this means we've achieved the major early phase program metrics of determining whether there is a there for the program hypothesis. And without going into specific details, because specific technical examples represent the intellectual property of the performers until and unless they publish those results. I can say we have collected sufficient data to suggest that several routes are plausible for the temporary interruption or slowing of complex biological events inside cells, as well as for the stabilization and protection of biomolecules from desiccation and denaturation. And we are moving into the phase of the program where maximizing the effect size and optimizing the effect translation become the paramount concerns. Any product designed for use in humans or animals will require extensive safety training, and the biostasis compounds are no different. If something is destined for use as a preservative, it needs to be tested to make sure it can be tolerated following dilution or reconstitution. And if it's a treatment in its own right, either alone or in combination with other therapies, then it also must be evaluated for appropriate dose and duration of the effect. Those questions, however, are beyond the scope of the current program goals. So, as I mentioned previously, Task 1 is essentially the program component that deals with the discovery, synthesis, and mechanistic exploration of our active chemical matter. That is, the things that interact with biological molecules and systems in order to produce our biostasis effects. We envision this program to be pursued much like a drug discovery effort, where a guiding principle is used along with some simple screening platforms to filter molecules from a large possible space down to smaller sets of hit compounds, exhibiting some degree of desired activity, which then is subsequently elaborated and optimized. For example, it's key to the issue of translatability that our intervention shows some degree of equivalent activity across a diverse array of biological models that can be interrogated quickly and in multiplex fashion. So think stabilization or reversible inhibition of multiple structurally and catalytically diverse proteins. Additionally, Task 1 incorporates the challenge of formulation for delivery. Not every intervention will exhibit ideal kinetic properties for diffusion, passive transport, et cetera, in its raw form. So we expect that the performer teams are going to investigate and optimize delivery methods and excipients to demonstrate the most robust and reliable formulation. Now, we use examples here of ingest, inject, or inhale, but in reality, the route of administration will largely depend on the interplay between the actual product mechanism, its form, whether it's a small molecule, polymer, et cetera, and the recipient system. So some products like biocompatible polymers may be most effective as acute interventions for wounds and would be formulated as a gel to be applied directly to the injured tissue and then stimulated to form a cross-link network, while others, such as small molecule chaperones or peptides, would be potentially administered in large volumes via intravenous routes to achieve the ideal distribution, kinetics, and concentration. These are hypothetical routes and activities. The program is still very much in its early phase of investigation. Which brings us to the other major technical task of the program. So you notice a theme running through my description of biostasis is the need to prepare iteratively determine how well candidate compounds work and whether or not their performance is contingent on the system it's being evaluated in. It is fully possible that mechanistic attributes that make for a high-performing product in one category or use case do not transfer to another. So to that end, the other technical task is to evaluate the degree of effect, its tolerability, and how well it performs in biological model systems of escalating complexity, and how consistent are the indications of the mechanistic underpinnings. So that means collecting data from multiple biological models across multiple conditions and perturbations to validate proposed mechanisms and the predicted performance from the task 1 molecules. This is also where the task 1 emphasis on formulation and delivery is actually applied and evaluated. And given that we have a diverse array of technical approaches, and that I wanted each approach to be evaluated routinely and repeatedly, every performer group is addressing both technical tasks. There is no outsourcing of empirical evaluation. There is no isolation of program components. In my view, it is critical to the success of this program that every development effort contains the capability to learn from the evaluation of their interventions and incorporate the results and lessons into subsequent optimization efforts. I have always thought that you learn more from a failure than you do from a success. There is nothing that ever works the first time it's tested. And there is often much to be gained from interrogating an appropriately conducted experiment, that's the caveat, that produces a null result. It is also a key component of the Biostasis program to maintain that empirically informed agility. If a particular route into cellular stabilization is successful but does not translate into a tissue or an organismal context, then we need to be able to re-vector the emphasis into a space where that validated high-performance attribute can enable a specific capability. What those readily accessible performance metrics are is ultimately what we use as an early indicator of research proof of concept being likely to translate into an applied technology. So this table here is a concise representation of our program metrics and model system platforms according to the program phases. So early on, we focused the efforts on those biological models and molecules that were most amenable to high scale and high throughput evaluation for effect and parameter determination, such as magnitude, concentration, response profile, et cetera. It was also critical that we begin factoring in tolerance of these early examples in at least a simple cell toxicity context. And you can imagine losing even just 5% of all the cells in a tissue or limb at once. That would be catastrophic. So ultimately, we want to get our interventions to the point where they are well tolerated by the system, regardless of what the system is. Moreover, we want to ratchet up the complexity of the models to reflect the translatability of the approach over time. The program is constructed in such a way that early on, the model systems and metrics are simple enough to facilitate large-scale screening of many, many iterations. Subsequently, enough should have been learned that a relatively more focused set of compounds and approaches are being evaluated in a more complex and thus more time-consuming model. To that end, the second phase requires not just demonstration of tolerance, but also the biostatic effect itself. And in the third phase, we're adding cellular community complexity. Tissues for protection can contain many different cell types, and we want to see how diverse the population can become and still demonstrate the same degree of effect shown in the more homogeneous models. And finally, we introduce a full physiological replication in the form of an organismal scale mammalian model system. The final phase of the program is the direct in vivo demonstration of a therapeutic application to prolong the survival interval of an acute challenge according to the mechanisms discovered and pursued along the course of the program. So these are direct quotes from the Biostasis program broad agency announcement that detailed how to assemble a demonstration of approach. It is an imperative in the program that satisfaction of phase milestones be met with a rigorous and reproducible demonstration experiment. So to that end, towards the culmination of each phase, the performer teams execute sets of experiments that are appropriately statistically powered and cover the content of the phase metrics for their approach in the agreed upon model system. Additionally, the components that are being evaluated are chosen in such a fashion as they demonstrate real world application, even though we are, as I mentioned before, operating in a truly exploratory and preclinical space. But for example, at the close of the first phase, where stabilization of simple molecules against temperature denaturation is a reasonable demonstration, we worked with the performer teams and took input from medical officers in the armed services to collect a list of field use protein-based therapeutics, such as various antivenins, enzymes like TPA, and antibody fragments, to use as test beds to provide tangible examples of how well particular approaches could perform if they were being used as novel protectants. And if we required quantitative measurements of resistance to stress, like heat or desiccation-induced denaturation, impacts on catalytic activity for an enzyme, or affinity to a target where it's an antibody, et cetera. And in this current phase, we're specifically looking at demonstrations for effects on human cells, including human blood components for performance evaluation, specifically addressing the known limitations to acquiring, transporting, and storing blood and blood products for field use. So this brings me to the end of all the material that I prepared for the seminar. I hope I've done a reasonable job describing the motivation for the program, its theoretical foundations, and the statement of testable hypotheses. And the directions we're taking the work as it navigates the capability demonstrations. So if all goes as planned, the program should yield a suite of interventions covering a number of potential applications. And perhaps more importantly, it will yield a de-risked fundamental perspective of how to approach complex perturbations to functioning biological systems and add new tools to the operator toolbox for preserving life amidst challenging conditions. So thanks very much, and I think we have time for some questions now.
Well, thank you very much for that presentation, Dr. Hey, it looks like I dropped off the net. Dr. McClure-Bagley, I think I'm back now. We did have a number of questions, and we'll go ahead and start getting on those right now. So from Michael Hauk, he wants to know, how would one reduce biostasis without also slowing the delivery or action of beneficial drugs?
That is an excellent question. And it's something that we are interested in pursuing once we get into that organ system level intervention. So this becomes a very interesting pharmacokinetic problem, where if you have a rate of onset of your stasis-inducing agent and your rate of reversal, we're operating under the hypothesis that that duration is what is necessary to basically prolong the functional capacity of the system under threat. And that once you start to reverse the process, if you do so in concert with the onset of another therapeutic, that the timing between those things is going to be relatively important. This could even get down to such things as the binding affinity for the therapeutic drug to its target if the orthosteric binding site of the protein target has been changed by the binding of the biostasis reagent. So it is something that we're definitely considering. But again, it's the kind of thing where I'm curious to see whether or not, first of all, the hypothesis is correct that you can actually prolong the intervention capacity for the system under threat. And if by tuning the on and off rates, you can successfully introduce the follow-on therapeutic without negatively impacting its activity.
Thank you very much. So the next question we have is from John Bowling. And he's curious, you talked a lot about the water content of the wood frog. And he's curious about if the water content of the wood frog versus the human do you perceive that going to be a significant factor in this?
So I consider water content to be a significant factor, mostly in the amount of water that needs to be potentially displaced from the hydration shell around the protein. So one of the things that is of particular value from a structural biology perspective in this program is understanding the relationship between immediate local viscosity and catalytic activity or conformational flexibility of the protein. And so, since every protein has a certain reliance on a hydration shell around it to facilitate interactions, both the hydrophilic and hydrophobic ones, because you have sometimes the buried regions in a protein, that are the more hydrophobic interactions, or in the case of transmembrane proteins, we have those hydrophobic coils. I'm curious about the interaction of the biostasis reagents and how that relationship unfolds. I'm not necessarily certain that the total water content of the cells is necessarily a determinant of the degree of protection once the biostasis state has been achieved. So it's an interesting question when it comes down to what is, like I said, the relationship between overall target protein hydration and the degree of reversible slowing. But beyond that falls into the category of extant differences between organisms that we have to sort of optimize around.
Okay, so Lawrence Roberge, you had a lot of questions on your slide 6. A number of people had queries on that. And he asked, if you do not allow genetic engineering, he wants to know if TDPs can be taken up via the cell membrane.
Yep, excellent question. And yes, in some cases, so that's one area. Again, I can't go into too many technical details because it's performer intellectual property. But there has been a lot of research, let's go for the last 10 or so years, on cell-penetrating peptides. So things that are capable of being taken up by cells either via stimulation of endocytosis or macropinocytosis, or just via, embedding in the membrane and then transiting passively. So that is part of the challenge for engineered protein design, is getting a relatively large therapeutic peptide cargo assembled in such a package that it can be delivered passively. There are other approaches as well, such as the formation of liposomal excipients. I mean, we're seeing a lot of that in the news right now with the Moderna and Pfizer vaccines. That's a huge part of how they work. Now, of course, it's delivering a nucleic acid cargo, but the same things can be used for therapeutic peptides.
Okay, so Mark Irvin, first of all, I complimented you on your presentation, Tristan. And then he went on to say that you can see the role of biostasis in reducing metabolic demand and prolonging negative cellular outcome. He said he wants to know if you've come across any measures that may protect cells without reducing cellular metabolic activity. For example, reducing the negative impact of acidosis and hypoxia without reducing the degree of local metabolic activity.
Very good question. And as of yet, okay. Again, I have to be careful because I can't talk about unpublished results. I can say that is a thing that we are looking towards. I can't speak with any direct certainty as to the potential, the mechanism that's still actively being investigated, but that is something that we are ready to see and interpret in the program. It is certainly possible. I will say that up front. It is certainly possible that a type of biostasis can be achieved wherein you either protect a threatened component of cellular machinery as a result of a cascading event downstream of a trauma that does not actually result from or involve a wholesale reduction in metabolic activity. That is certainly possible. And there are examples of that actually in the literature. I'm not sure to what extent the model has been translated. There's a group at the Hutchinson Cancer Institute that has done some work with elemental reducing agents in protecting against reperfusion stress following trauma. Mark Roth, I believe, is the PI who's published the majority of those papers, but that is certainly a thing that is possible.
Great. So we had another question, and from Joe Hack, and I think Joe has Joe works forward with our combatant commanders, and you know, as you and I talked, Tristan, as we were working up to this presentation. My experience in Iraq when I worked ops in Al-Anbar is we were greatly limited in where we could go because we didn't typically conduct operations outside the proverbial golden hour. So if we couldn't medevac a wounded service member back to a higher level of treatment within an hour, then we were not going to do that operation typically. And I think that, you know, these technologies, the ability to extend that time period, I can tell you from practical experience when the MV-22 Osprey was deployed during my tour in Al-Anbar, it extended our reach because it could get medevac wounded so much more quickly. And Joe asked kind of a follow-on to that idea. He's curious if you thought ahead to how the technology would be integrated in trauma. Is it something you think that perhaps, or you desire that a corpsman or a medic would carry with them and be able to administer in a more austere environment, awaiting such an air medevac or ground medevac? What have you thought through how you envision this being used in both civilian trauma as well as in military operations?
So yeah, that's a great question. Thank you. I think it's probably best if I answer in order of where I think general technology readiness is. We have one effort in the Biostasis program. The research is taking place at the University of Wyoming in collaboration with the company Cellfire. And their entire goal is to generate a more stabilized whole blood product. I think that that kind of technology is what is likely to be, I could be wrong, but I think that is the type of application that is the closest to a higher technology readiness level, and as such would represent the kind of thing that you would imagine would be used, by a medic. Anyone who would be trained to administer an IV or administer blood product, you would then have access to that type of intervention. And as such, you would use it under the identical circumstances. You're just changing the availability of that product. When it comes to the other types of interventions, again, I could offer an opinion, but it would be a completely unsupported opinion because I really want to see more of what the ultimate validation of the hypothesis looks like in the organismal level model systems, right? If it becomes a thing, as the first question alluded to, is it something that interferes with the action of another therapeutic drug if it's being administered? Well, then that requires a lot more, complex monitoring and dynamic, maintenance of the recipient than if it's something that can be delivered as a simple adjuvant, or adjunct therapy along with, rehydration or blood product, et cetera. So in order of application, I think what you're first likely to see are stabilized field forward use of blood products, and then second would be the more physiological supportive capacity. And I think that those two things probably also carry a little separation in terms of the training required to administer them.
Super. I think that nailed it, Dr. McClure-Begley. So That's all we have time for today, and I'm very grateful for your presentation. It was obviously very well received with the number of questions that we got today. And I'd like to thank everybody who joined us today. As I said, we live in challenging times, and the holiday season always puts demand on all of our time, so I'm very grateful for all of you joining us today. As mentioned at the beginning of today's webinar, the slides and recording of this presentation are going to be available for download at the HDIAC website. Several of you had questions about that, and we will absolutely make those available to you. If you're interested in learning more about the IACs or getting involved as a subject matter expert, presenter, or expanding your presence in our user community, please feel free to reach out directly, and you can use the contact information you're going to see appear on the left-hand side of the screen. In addition to these monthly webinars, the HDIAC also offers monthly podcasts, bi-weekly email digests with the latest scientific and technical news, as well as a technical inquiry service where we offer up to 4 free hours of technical support and consulting. And we can also perform customer-funded work in any of our technical areas via a pre-competed contract vehicle. So again, that information is included on the left-hand side of your screen in the chat box. Please be on the lookout for our January podcasts. We're going to be hosting Dr. Stephen Thomas from the State University of New York Upstate Medical University, where he's the chief of the Infectious Disease Division. We're looking to do that on January the 15th, and he's going to be talking to us about infectious disease impacts to the warfighter. And additionally, we're going to be hosting Ambassador Rich Kozlerich, who currently works at George Mason University Schar School of Policy and Government, and he's going to talk to us about energy security. And we're looking at doing that one probably on the 21st of January. Both of these promise to be great events, so be sure to register on our website to receive notification about all of these product releases. Again, Tristan, thanks so much for that presentation. Thank all of you for your time today. Like us and follow us on social media as well. And best regards for a happy holiday season. Please stay well. Thanks.